Hello, everybody. I guess we better make a start. So, how's your year been so far? Everything all right for the beginning of the academic year? Yeah. I'm glad you said that because I had no intention of solving any problems you may have had. So, it's just as well that everything is going fine. Okay, so you're going to be having general microbiology with me, Mr. Sacagnon, and Madame Mouratou. And I think this is correct for the way we're going to organize the courses. So they are all put down in your timetable as a CTD, kind of supposed to be mixed lectures and class teaching. But in fact, we don't really work like that. We try and do the same kind of things we do for the other L2 microbiology students. So there's a lecture part, that's me and Mr. Sacagnon, and there's a kind of class teaching exercises part, which will be done by Madame Mouratou. So basically, you start off with me for the first few lectures on bacterial structure and function, a little bit about bacterial taxonomy, and then we go into nutrition and energy metabolism. And then Mr. Sacagnon will take over with Madame Mouratou for the next few weeks. And then you'll see me again after the autumn break for something on bacterial pathogens and bacteriophages and viruses. Probably most lectures are going to be about the same as in previous years, apart from probably these ones. I plan to change these a little bit. So I'm just thinking actually, have I put up the PowerPoints of the lectures yet? So on Madoc, you should have the PDF files of all these slides. And I'll also be recording the video of the lectures, and I'll put them up on YouTube so you can listen again to any sections where you didn't quite catch what I was saying, or you want to make sure you understood correctly. Okay, so you will have access to those as well. However, probably these ones, these lectures here, I'll put up the slides maybe just about the week before the lectures. Okay, and actually I shall check this evening to make sure I have these ones up. And if I don't, I'll put them up today or this evening or tomorrow. That's what we do in the second year. If you are interested in microbiology, you can take the virology, mycology, parasitology option in third year. And you can also carry on and do microbiology, or at least some parts of microbiology at the master's level, because in the M1 we do an applied microbiology course, and in the BBRT M2 there's an infectious diseases option that you can take, and this is still going to exist from 2017 to 2022, so it will still be available for you. Now, for reference works that you might find useful, pretty much any standard microbiology textbook has the material that we cover in these lectures and in this course. So I pretty much base my work on the Prescott, Harding and Klein's microbiology, but Perry is also very good. Any other microbiology textbook is great. This is a kind of free online microbiology textbook, mostly aimed at medical students, and the bacteriology section has not yet been entirely translated into French. But the idea of this project is to have the same online textbook available in different languages. So the main one is in English, but it's also been translated into a lot of other languages. Microbe library, this has got or used to have a lot of images and resources from the American Society of Microbiology, but it's moved aside, so this link doesn't work anymore. And this is a link to a microbiology blog run by a retired professor who posts a lot of interesting stuff about microbiology. Could be bacteriology, parasites, viruses sometimes, but it's very, very interesting, and this is one of the blogs I follow for microbiology. Okay, so in terms of what we do in each lecture, okay, this is not really to talk about, but you'll have it in the slides just to, if you want to find a bit more about, I don't know, structure of the bacterial cell. These are the sections in the different chapters in Prescott where you can find the relevant information. Yeah, okay, so I don't need to say anything more than that, really. Now, what we're going to be talking about in this course is microbiology. And there's a very, very simple definition of what microbiology is. That is, it's the biology of organisms you cannot see with the naked eye. So anything that's too small to see, that's microbiology. So it covers microscopic eukaryotes, you know, this kind of very, very large taxonomic category called protists, which is very diverse. It is not clade in the phylogenetic terms. Yeast and fungi, so what you might call unicellular eukaryotes. And then you've got prokaryotes, which are divided into eubacteria, the true bacteria, and archaebacteria. And as well as that, you've got viruses, which are not cellular forms of life. Now, what we are going to be focusing on in the course is mainly bacteria. Okay, so it's mostly bacteriology, and within bacteriology, mostly we're going to be studying eubacteria. There'll be one lecture on archaea. Now, that's not because these guys aren't important, but we have only a limited amount of time, and we think we should focus on bacteria to start with. And there'll be 2 lectures on viruses. So these aren't covered, but nevertheless, they make up part of microbiology, and we can cover some of these in the 3rd year course with the mycology, parasitology. Please come in. Okay, so we're mostly focusing on bacteriology. Why? What's so great about bacteria? And I'm not a bacteriologist, but every time I come back to this course, I have to remind myself and rediscover some interesting things about bacteria. And it's always a pleasure for me to come back to bacteria. Now, they're important to study because they're everywhere. In every single environment on Earth, you can find bacteria. You can find bacteria, you know, where you expect to find them, you know, in water, in sewage, in the soil. You can also find bacteria in lots of unexpected places. In the Antarctic, in the bottom of a frozen lake, there are still bacteria. There are bacteria in boiling hot springs at 95 degrees centigrade and pH 2. There are bacteria all over your body, in your mouth, in your skin, over on all of your mucosal surfaces. And in all of these places, they're doing something useful. Well, yeah, mostly. And they can live anywhere because they can live on practically anything. They can get metabolic energy out of, well, from a surprisingly wide variety of sources. Normally, when you think about metabolic energy, okay, thinking about macroscopic organisms, animal life, plant life. Animals, they need to eat something. They need organic chemicals. You eat them, you get energy. Plants perform photosynthesis. Well, bacteria do these things, but they can do a whole bunch of other things as well. And they manage to do that by having all sorts of clever biochemical pathways. They can obtain metabolic energy from, just oxidizing iron 2 to iron 3. You can get enough energy out of that to grow. This is quite an amazing thing that bacteria do. And because they can perform these sophisticated, unusual, difficult biochemical reactions, they're very useful in a variety of industrial applications. So, of course, the big ones are things like, making cheese, yogurt, okay? And then they're useful for making antibiotics or some other transformations, making recombinant proteins in Escherichia coli. So industrially, they're important. Another reason bacteria are interesting is because they're the simplest living organisms. So if you're interested in the fundamental processes of life, DNA replication, protein translation, then bacteria are the simplest model organisms to look at these processes. And from a more human-centered view, there are many very important bacterial diseases, not just in the past, but also in the present. And it's very important to study bacteria in order to be able to treat these diseases and cure people or prevent these diseases or cure them. And last but not least, although perhaps from a biomedical point of view, most of people's attention is taken by pathogenic bacteria, most of the bacteria that live on or inside the human body are not harmful at all and are probably very important for the normal functioning of our digestive system, keeping a healthy skin, keeping a healthy mouth, this kind of stuff. And we're only beginning to understand the roles of these commensal bacteria in, keeping a variety of our physiological functions running correctly. So, there's a lot of interesting work going on now to try and find if there is a link between healthy gut bacteria and obesity or diabetes or different problems in the immune system. So this is a new way of looking at the impact of bacteria on human health. So those are the reasons why bacteria are interesting. Now I'm afraid we go right back to the beginning and look at the basics of bacterial structure. So I'm going to take a little bit of time in the next couple of slides just to try and outline how we know this stuff, okay? Because afterwards I'm going to explain and present, there are different types of bacterial structure. They're like this, their function is that, and this kind of stuff. But it's worth having an idea how we gained all of this knowledge. Well, for a start, the simplest way to have an idea about bacterial structure is just by looking at them down a microscope. And of course, bacteria are big enough to be seen by light microscopy. Now, if you're very lucky and you have a nice phase contrast microscope, then you can quite clearly see bacterial cells without having to stain them or color them in any way. So you can see the size and the shape of these cells. And sometimes you can see these are examples of inclusion bodies in these bacteria. So you can see that some bacteria have some kind of structure inside them. Now, most of the time, you won't have a phase contrast microscope. You'll just have a simple light microscope. And in order to see bacteria clearly, again, you'll have to stain them up and color them with something. So there are simple stains, which will pretty much color every bacterial cell and do not differentiate different types of bacteria. So crystal violet, methylene blue, these are simple stains. So once again, you can see the size and the shape of bacteria. You can see some structures, like around these kind of cells, it seems kind of indistinct. There's a kind of light blue halo around these cells. That's probably capsule around this outside of the bacterium. So simple stains have some kind of utility. You can see whether you've got bacteria or not. But what are most commonly used are differential stains, which give you some kind of information about the identity of the bacteria. So the most common is gram staining. So gram positives are this purple color, gram negatives are pink. And I hope in the lab classes you'll get images just as nice as this one. Another type of differential stain is acid-fast staining. That's specifically to stain mycobacteria, so for tuberculosis. Still the most commonly used diagnostic test is you get someone to spit out on the slide and you stain the bacteria, stain the sputum for acid-fast bacteria. So these are mycobacteria which will specifically retain the stain despite the very stringent washing conditions. And then some stains are specific for particular structures inside the bacteria. So this red stuff here is specific for endospores. Okay, so let's go back to gram staining. What kind of information does gram staining give you? Well, it tells you if a bacterium is gram positive, in which case it will be dark purple, or gram negative, in which case it will be pink. And you can see the shape of the bacterial cell, which will of course be, not all the time, but mostly either spherical cells, cocci, or rod-shaped cells, which are bacilli. And for example, cocci, you can divide them into different types of cocci. Monococci, they're on their own, diplococci, staphylococci, and a kind of cluster or in clumps, and streptococci in a strip, in a line. So rod-shaped bacilli have different forms as well. If they're very, very long like this, you would say, okay, they're bacilli. If they are comma-shaped, curved like this, they would be called vibrio. And if you have a kind of very short oval-shaped cell, this is kind of, you might call it cocobacilli or coliform shape, because Escherichia coli looks like this. So other bacteria that are gram-negative with this shape are coliform. That's not the only shape. Yes? The question is, what is gram-negative or gram-positive? Actually, I'm coming back to that, but, okay, if you perform this differential staining, some types of bacteria will stain purple. They're gram-positive. Others will stain light pink. They're gram-negatives. And this is because of the composition of the cell wall, which we'll see later. Okay, these are not the only forms of bacterial cells, in fact. So sometimes you can have also spirochetes, which are kind of like wiggly spaghetti pasta or something like that. Okay, there's another common form. But mostly, okay, just stick to coccibacilli. Yeah, just to remind myself, not all bacteria can be classified as gram-positive or gram-negative. Okay, so the acid-fast bacteria, mycobacteria, you can't differentiate them with gram staining. You need this other type of staining to identify them. And some bacteria do not have a cell wall at all, mycoplasma. So they can't be classified as gram-positive or negative either. So light microscopy, the first way to identify something about the structure of the bacterial cell. Of course, light microscopy is limited by its resolution. So if you want to see structures that are smaller, then you need to use electron microscopy. And you can either use scanning electron microscopy to get an idea of the surface of the bacterial cell, or transmission electron microscopy. And then there are two different techniques that are used here. This one is by taking a very, very thin section through the bacterial cell. Okay, so it's cut across the middle. And then you can see the structure inside the bacterium. The main thing that you can see is that it's not very complicated. Okay, you've got the cell wall. You can see the DNA here in the kind of nuclear body. And here you've got an inclusion body. So you can get some kind of information about the structure inside the cell. Another thing you can do with transmission electron microscopy is kind of shade the surface of the cell with carbon and platinum, and then use that to give you a kind of imprint of other structures on the outside. If you do that, you can see that, in fact, many bacterial cells are not just, smooth capsules like this, smooth lozenges, but they have a variety of structures on the outside. So this big long thing here is a flagellum, which normally cannot be seen by light microscopy because the diameter is too small. And then you have all these small filaments around the outside of the cell, which are called pili, which means hair, right? Poilu, pili. So these filaments, pili or fimbriae, are on the outside of many bacterial cells, gram-positives and gram-negatives. So the surface of the bacterium is rather more complicated than what you might think just by looking by light microscopy. Here it looks all smooth and very, very simple. If you use a higher resolution, you can see that the surface is much more complicated. And the last approach to get even more detailed information about the structures in bacterial cells is, well, for example, the biochemical approach. You might be able to identify a structure like a flagellum on the outside of the bacterial cell. And you say, okay, we'll smash up the bacterium, separate it into the different parts. We'll try and purify the flagellum on its own. And then we can analyze it biochemically and see what it's made out of. So this is how we know what the chemical composition is of different structures in the bacterial cell. Like this thing here, lipopolysaccharide. It's on the outer leaflet of gram-negative cells. And we know exactly what its structure is. You've got a kind of lipid part here that's forms the outer membrane. Core polysaccharide, and then the kind of variable polysaccharides on the top. That's one way of getting information about structures. Another one is to try and combine genetics, biochemistry, and structural biology. So if you've got a structure on the bacterial cell and you want to find something out about it, you can try and generate mutants that don't have this structure. Then you try and find the gene that's mutated. And once you've got the gene that is responsible for making that structure, then you can clone it, try and resolve its 3D structure and find out how it works. So an example here are the membrane porins and permeases. These are in the outer membrane of gram-negative cells, and they all form kind of trimers of this beta barrel structure. So the outside here is all hydrophobic, so it sits in the membrane. But if you look at it from the top, you can see that it forms a big hole through the membrane, and hydrophilic molecules can pass through here. And at the end of it all, if you combine these approaches, electron microscopy, genetics, structural biology, and biochemistry, then you can build models and understand how these things function. So this is an image of the basal assembly of a gram-negative flagellum. And it's not just a kind of artist's impression. This is a real model of what it is really like. Every one of these things is a different protein. And when there's a line, these are two different copies of the protein. So in terms of size and number, this is absolutely true to real life. Okay, it's a real view of what it's like. And this is something that's been built up by studying this thing over, you know, decades. Okay, so let's see if you've been listening. So I'm going to run this with mClicker, just for fun, I guess. Should be simple. So if you've got your mobile phone or your computer, you can get into mClickers, and I will send out the question. So if you connect to this site here, it'll ask you for a session code, and it's this 292481, and then you can send me your answers. Okay, so let's see what we got. Okay, those are the answers from the 11 people who answered. Okay, number one, capsule. Yeah, everybody got that right. Yes, it can be seen with often with a simple stain. Pili LPS, of course, cannot be seen. Pili are too small. LPS is just an organic molecule. Nucleus for people. Hey, look, I said that's why I corrected bacterial structures. Bacteria, they don't have a nucleus, right? So this was a, no bacteria have these. Endospore, only like, what is that, one person said yes? No, you're right. Endospores with a differential stain, or even without, you can often see endospores inside a bacterial cell. They're big enough. Okay, so okay, so the nucleus. Don't forget, no bacteria has a nucleus, right? So this gives me an excuse just to give you a quick reminder of all the differences between eukaryotic and prokaryotic cells. So, of course, eukaryotes have organelles, sophisticated membrane-bound internal structures that carry out important functions. Bacteria, they don't have real organelles. Some bacteria have inclusion bodies. These are just for storage. And some of them can be membrane-bound. But these membranes are not very dynamic. Okay, they're not doing a lot of interesting things like the membranes in eukaryotic cells. Eukaryotic cells have a very extensive cytoskeleton that keeps the cell in a not exactly rigid, but at least, structured shape. Tubulin and actin were thought to be totally specific to eukaryotes, but in fact there are some homologs in bacteria that play, well, okay, not exactly the same roles, but you can see some kind of similarity in what FTSZ and MREB are doing in bacteria compared to tubulin and actin in eukaryotes. Cell wall, okay, some eukaryotes have got cell walls. Let's say most bacteria do, almost all. Chemically, they're not the same kind of thing at all, though. So this is convergent evolution. They're not homologous structures. And we can say the same thing about flagella, okay, so can be present in eukaryotes and prokaryotes, but very, very different structurally. So they are not homologous. Okay, external membranes, only one in eukaryotes, one or two in prokaryotes. Gram-negatives have an external membrane and mycobacteria. Pili are specific in bacteria. No eukaryotes have them. Yeast don't have them. Human cells, plant cells don't have them. Spores, yes, some eukaryotic cells have them. Once again, similar function, but not at all similar structures. So these things, spore, flagella, cell wall, similar functions in some eukaryotes and in prokaryotes, but they are the products of convergent evolution and they are not derived from a common ancestor. Now, in terms of genomes, there are very, very many important differences between eukaryotes and prokaryotes. Of course, the most important being the presence of the nucleus in eukaryotes. Okay, so transcription and translation are physically separated in eukaryotes, and they are not in prokaryotes. So in eukaryotes, the genome is diploid at least at some stage of the organism's life cycle, whereas all prokaryotes are haploid. Eukaryotes, well, you have several, and in general they're linear, whereas most prokaryotes have just got one circular chromosome, and accompanied often by plasmids, whereas you don't have any plasmids in eukaryotes. And another big difference is the presence of histones in eukaryotes, which are absent in prokaryotes. Some exceptions. Okay, some bacteria have two circular chromosomes, and some archaea have histone proteins, or let's say DNA-associated proteins that are very, very similar to histones. Okay, So you put all that together and gives you this kind of idea of the structure of a typical bacterial cell. A variety of structures on the outside, on the inside not very much. Okay, nucleoid, cytosol, some inclusion bodies. Now what we're going to do for the rest of this lecture and maybe the next one is to go through these different structures, look at what they're made of, how they're produced, and what their function is. And I'm going to go from the outside towards the inside. Okay, so we're going to start with the external structures, do them first, and then do the internal structures next time. So I'm going to start with the cell wall, okay, because it's very important for this gram staining stuff. Now, what is the function of the cell wall? Very simply, it's going to give a rigid structure to the bacterial cell. And that's important because it helps the bacterium to resist osmotic pressure. Because otherwise, if you put bacterium in some kind of dilute solution, of course, water will enter the cell by osmosis and the cell is going to explode. That's what would happen with your red blood cells if we put them into a glass of water, right? They would all lyse. So bacteria, the main function of the bacterial cell wall is to prevent this from happening. Now, just on this subject, I don't want to say it afterwards. Now, the structure that provides this rigidity is the same in all bacteria. It's peptidoglycan, which is a very conserved structure. It's specific to eubacteria. Okay, archaebacteria don't have peptidoglycan. They have other polymers. Might be a little bit similar, but it's very specific to eubacteria. And so, eukaryotes don't have it. And this can be used as part of our body's defense mechanism. So many animals secrete lysozyme. And this enzyme can specifically degrade peptidoglycan. So you have it, in your tears, maybe a little bit in your saliva. It's secreted by macrophages and it can destroy the bacterial cell wall. And peptidoglycan is also a target for different families of antibiotics, including the beta-lactams, okay, penicillins, cephalosporins. Okay, and it's historically been a very important thing to know about bacterial cell wall structure because differences in cell wall structure are what determine whether bacteria are gram-positive or gram-negative. So just to explain what this means a little bit, everybody is going to do gram staining in the lab classes, and you really need to know this by heart, okay? How does gram staining work, and what are the differences between gram-positive and gram-negative cells? So, as we'll see in the lab, firstly, the first step in gram staining is to stain the cells with crystal violet. So initially, this is a basic dye. It enters all the cells and will form complexes with nucleic acids, for example, something that's got a negative charge inside the cell. Then you treat the stained cells with iodine, which is a mordant. So this is going to form large complexes of crystal violet and iodine in a big kind of like clump, which do not diffuse out of the cell easily. Then the third step is to wash the cells with alcohol. So this is a decoloring agent. Some of the cells will give up their crystal violet stain and some will retain it. So at this stage, gram-positive cells are purple and gram-negative cells are entirely invisible. And the last step is to counterstain with a fuchsin, and this will stain the gram-negatives pink. Now, so this is the basic difference between gram-positive and gram-negative cell walls. gram-negative cell walls can be dissolved with alcohol, and gram-positive cell walls are relatively resistant. Okay, so most bacteria can be characterized as either gram-positive or gram-negative. Most, but not all. One example are the mycobacteria. They have a distinct cell wall structure. They've still got peptidoglycan, but they have an outer membrane, and it's a little bit different. It's not the same as a gram-negative cell membrane, outer membrane. And you need a specific cell staining protocol to identify mycobacteria. That's acid-fast staining. And there are some bacteria which don't have a cell wall at all. These are mycoplasmas. And you might imagine, why is it that they can survive without a cell wall? We started off by saying it's very, very important. It needs to protect the bacterium from osmotic pressure. How do these guys survive without one? Well, the mycoplasmas are parasitic bacteria. They always grow together with a eukaryotic host. So they grow on the surface of eukaryotic cells. And from those cells, they recover cholesterol. And cholesterol has the same function as in a eukaryotic cell membrane. It makes it more rigid. So these bacteria manage to keep a rigid rigid cell membrane by using cholesterol from their host cells. And they can do that because of their kind of parasitic lifestyle. So that's not an option which is available to most other bacteria. Okay, so for most bacteria, a cell wall is essential. Okay, so gram-positive cell walls resist alcohol. Gram-negative cell walls are sensitive to alcohol. And if you're lucky, you can look at the structures of these cell walls by electron microscopy. And what you see is that in a gram-positive cell, so this is the cytosol, this is the outside, you just have a plasma membrane and a thick layer of peptidoglycan. Whereas in a gram-negative cell wall, you have a plasma membrane, a thin layer of peptidoglycan, and an outer membrane. So in a gram-negative cell wall, you've got a lot of lipid and a relatively small amount of peptidoglycan. In a gram-positive wall, just peptidoglycan. So alcohol is going to dissolve this outer cell wall, and the peptidoglycan is quite thin here, so it will be permeable to the dye and it can escape. Whereas what seems to happen with a gram-positive cell wall, when this is treated with alcohol, the peptidoglycan layer will kind of become dehydrated. It will collapse and become less permeable. Okay, peptidoglycan. This is very important. This is what is present in gram-positives and gram-negatives. It's the part of the cell wall that allows it to be rigid. You know, it's like a Kevlar bulletproof jacket or something for bacteria. So what is it made of? Peptidoglycan. Okay, the clue is in the name, right? Peptido, you have some kind of peptide. And glycan, you have some polysaccharide. So the polysaccharide repeating unit are dimers of N-acetylglucosamine, which I'm going to call NAG from now on, and N-acetylmuramic acid, which I'm going to call NAM. So the polysaccharide bit goes like this. It goes NAM, NAG, NAM, NAG, NAM, NAG in a big line, okay? Now, that's not very useful to give you a rigid structure, okay? You've just got a linear polymer like this. It's like you've got a whole load of threads. In order to make this rigid, then you've got to cross-link the different threads, okay? And that's the function of this tetrapeptide. The peptide part, the role is to cross-link the different NAM-NAG polymers. So the tetrapeptide is on position 1, 2, 3, 4 here. And it's very unusual. What it's got, have a look at these amino acids here, okay? What is unusual about these guys? Two things. Anybody spot one? What is this? Alanine, glutamate, right? Alanine. Okay, we recognize these, the amino acids. This thing, what is that? It's unusual, okay? It's not an amino acid that you find in proteins, this one, diaminopimelic acid. What's the other unusual thing? What does that mean? L, D. No? Anybody? Hmm? Yeah. Yeah, so they have different chirality. What are the ones that you find in proteins? What amino acid do you have? L or D? Hmm? D? Everybody says D in proteins. Yeah, it's L amino acids in proteins, right? Yeah. And D for sugars. D glucose, this kind of stuff. But it's L amino acids that you have in proteins. So here you have these D amino acids that are involved in this tetrapeptide. So this is unusual. It's an unusual structure. So right away, this is telling you this is not synthesized on a ribosome. It's not like part of a protein that's been cleaved off or something like that. It's being produced in a different way. Okay, so this third one, diaminopimelic acid, in this position 3. I don't know if we might see its structure a bit later on, but the important thing that it has is that it's got an amine group on the side chain. And it can form a kind of peptide bond between the carboxylate group at the end of this D-alanine. Okay, so they're numbered N-terminal to C-terminal here, just like in a protein. So this is the N-terminal, and this is the C-terminal. So this has got a free carboxylate. And the amine group from the side chain of diaminopimelic acid can form another peptide bond here with the carboxylate at the end of the D-alanine. So you have these peptide bonds, these trans peptide bonds that are going to cross-link the chains of NAM-NAG, the glycan polymer. And that's what holds the whole thing together. Now, how is this synthesized? This is not a trivial question. Okay, it's a very difficult thing to imagine how bacteria do this. Well, we'll see. We'll see it's complicated and try and understand why it's so complicated afterwards. Okay, so the main steps are okay, synthesize the repeating unit. Okay, the repeating unit is what? The repeating unit is this NAM-NAG disaccharide with the amino acid stuck on it. So first, in the cytoplasm, the repeating unit is synthesized. The NAM-NAG pentapeptide, and it's attached to this lipid transporter, which is called bactoprenol. This flips in the membrane. So the repeating unit, the NAM-NAG pentapeptide, is going to be outside in the periplasm. And then it can be inserted into the cell wall by transglycosidases. And the peptide bridge is is formed by the action of transpeptidases. So that's the summary. Now, up until last year, there was very nice animation here on the one of the American Society for Microbiology websites, but the link was broken when I checked it this morning. So I'm going to try and comment this YouTube video, which I think, for me anyway, shows the biochemistry involved in a very pertinent way. I'm going to bring this over here. Okay, so we start off with NAM, okay, N-acetyl-muramic acid. And the very first step is to kind of activate it by combining it with uridine diphosphate, UDP. Now, NAM, NAG, which ones are the peptides fixed to? I always have a trouble remembering this, okay? But you've got to remember that you want to form a kind of peptide bond. So NAM is acid, N-acetylmuramic acid, so it's got this carboxylate group. This is where your peptide's going to be fixed onto, okay? N-acetylglucosamine doesn't have that. It's got an amine group. It won't work, okay? So the peptide is always going to be on the NAM sugar, N-acetylmuramic acid, because of this acid group here. So what's the first step? Let's have a little look here. So the first step is going to be fixing on the first amino acid, which is what's D-alanine, right? Probably that's the guy on the soundtrack saying what I just said, but in a slightly more organized way. Okay, right. So you get L-alanine first. That's the first amino acid that goes on here. So you've got peptide bond here, right? And the next one's put on glutamic acid, glutamate. But every time you form a peptide bond, what do you need? Is this reaction going to work on its own? Yes or no? Got a 50-50 chance. Who says, yeah, it's going to work, just going to go? Anybody? Who says, no way, that'll never happen on its own? Nobody. Oh, that means you don't care, right? Well, you should care. You should care. Because you have to understand how these processes work. Peptide bond formation takes energy. It's not a favorable reaction. Why? Anybody have any idea? I know we're coming at the wrong time of year because you haven't done biochemistry yet. Right, look at this bond up here, okay? Before. What are we getting rid of here to make this bond? Water, right? Water is the leaving group. If you're in an aqueous solution, this is going to be highly unfavorable. Okay, even in the bacterial cytosol, there's a lot of water around. To expel water in an aqueous solution is going to be very, very unfavorable. Just in a simple way, okay? I don't think I'm a fantastic organic chemist or anything, but this is quite a simple idea that at least I can grasp. So this kind of condensation reaction is always going to be unfavorable, right? And the same is for a glycosidic linkage. You have a water as a leaving group. Okay, so energetically it's going to be unfavorable, and you always need to put energy in to make these anabolic reactions. But you're in the bacterial cytosol here, you've got ATP. Every time you put an amino acid on here, it takes ATP. Okay, but that's okay, you've got ATP. Okay, so the first 3 amino acids are put on one by one, stuck onto the N-acetylmuramic acid. So the diaminopimelic acid comes on, and the last one is a little bit different. This dimer between 2 D-alanines is synthesized first as the dimer, and then the dimer is stuck on at the end. There's a very, very peculiar biosynthetic pathway and very, very specific. Bacteria do it and we don't do it. So what you've got is this UDP NAM pentapeptide structure synthesized in the cytosol, and then it's going to be attached first on the inner leaflet of the cell membrane, and it's going to be attached onto this transporter, bactoprenol. Okay, so you get rid of the UDP phosphate, and the phosphate group is attached onto the bactoprenol. So you've got this bactoprenol diphosphate NAM pentapeptide structure. And then finally, UDP N-acetylglucosamine comes along. Once again, the UDP gets lysed, and it's the energy from this phosphate group that is used to produce the glycosidic linkage here. So this is what you get produced on the internal surface of the bacterial cell membrane. Then this whole structure is going to flip in the membrane. So you get bactoprenol diphosphate NAMNAG pentapeptide on the outside. Then transglycosidases can bind this, can add it to peptidoglycan. Let me just stop that there. Why is this going to work? Because you're cleaving the phosphate bond here. So you get some energy from cleaving this bond. That's what allows you to add on to the polymer. Okay? And because this is stuck in the membrane, doesn't just float away into the, outside the bacterium doesn't lose bactoprenol, it's stuck in the membrane and it gets recycled. Okay, with its transport, it will lose one phosphate, so that's where the energy comes for the transport. And then the last step is the transpeptidase reaction. So the transpeptidase is going to form a peptide bond between the amino group on the diaminopimelic acid and the carboxylate group of the 4th alanine. So what is happening here is you form one peptide bond and you break another one. The 5th amino acid is removed. So that's why it can work outside the cell. Otherwise, you would need ATP and an enzyme to perform this, to make this peptide bond. But you don't have any ATP, you're on the outside of the cell, right? And it can work because you've produced a pentapeptide and you use the energy in the last peptide bond to produce these transpeptide links, these cross links. Okay, so that's why this whole process is so complicated. The big problem for the bacterial cells is how do you produce a complex structure on the outside of the cell? Because outside of the cell, you don't have any ATP, you don't have any NADPH, you don't have metabolic energy, right? So you've got to produce things on the inside of the cell in a kind of activated form, export them, and then use the energy that you've kind of like locked up in the pentapeptide to produce the final molecule, the final polymer. So that's all very clever. Well, I think so anyway. Okay, so the last thing about this is that it's a very, very important, highly conserved biosynthetic pathway that almost all bacteria use. And that's what makes it a very interesting target for antibiotics. Okay, so penicillins and cephalosporins, these are beta-lactam antibiotics, they inhibit the transpeptidase activity. So the last step in this process can't work. And in fact, the transglycosidase and the transpeptidase activities are linked. Okay, so in the video, it looked like it was one process and then the other one, but you know, it's a big enzyme complex. It does both reactions one after the other. So if you block the transpeptidase activity, you're blocking the whole system. Okay, so the whole of the cell wall synthesis is going to stop. And then bacteria can't resist osmotic pressure, they die. Vancomycin, also an antibiotic that targets cell wall synthesis. So these ones, penicillin and cephalosporins, can be effective against gram-positives and gram-negatives because they both need to produce peptidoglycan. Vancomycin is only effective against gram-positives. It has a different mode of action. What it does is it binds very specifically to the D-alanine dimer. So when the NAM-NAG bactoprenol pentapeptide comes on the outside of the membrane, you've got this D-alanine dimer. So before the transpeptidase breaks it down, vancomycin will bind to the D-alanine and also block this enzyme. And another antibiotic that works here will prevent the translocation of bactoprenol. So it's going to stop the transport of these NAM-NAG pentapeptide subunits across the membrane. So that's bacitracin will inhibit bactoprenol, but it's too toxic to be used systemically. It can only be used locally, even though it's very effective against bacteria. Okay, so that is synthesis of peptidoglycan, which is present in gram-positive and gram-negative cell walls. However, there are some differences between gram-positives and gram-negatives. So Escherichia coli, a typical gram-negative, it will have this diaminopimelic acid in the third position, and the cross-links are direct from one NAM-NAG polymer to the next. Staphylococcus aureus, typical gram-positive, has lysine in the third position, not diaminopimelic acid, but it's got the amine group on the side chain, right? That's the important part. And the peptide bridge can be, you know, a polymer of glycine. So this interbridge can be longer. So why is that important for gram-positives? Because overall, in gram-positive bacteria, the peptidoglycan layer is much thicker. So it's important perhaps to be able not to just to link neighboring NAM-NAG polysaccharides, but perhaps to make the interpeptide bridges a little bit further along in this structure to keep the whole thing together. So gram-positive cell wall, very, very simple. Okay, you've got the plasma membrane here with integral membrane proteins. You'll have a periplasmic space in between the plasma membrane and the peptidoglycan layer. And the other thing you have is teichoic acids. So these are kind of repeating structures like this of ribitol or glycerol phosphate. So this is glycerol phosphate repeated. Can be ribitol, so 5-carbon sugar. And R is going to be some kind of sugar or an amino acid. Could be D-alanine or glucose or something like that. So what is the role of these big polymers here? So they've got a net negative charge with all these phosphate groups, and they all have electrostatic interactions with positive charges on the peptidoglycan here. And so they help, you know, keep all of these layers of peptidoglycan together. It's just a structural function. Some of them are anchored in the plasma membrane, so this is lipoteichoic acid. Just one more slide, and then I'll give you something to think about, and then that'll be the end for today, I guess. Gram-negative cell walls are more complicated. So you've got plasma membrane, integral membrane proteins, the periplasm in between the plasma membrane and the peptidoglycan layer. It's thin, And on the outside, you have an outer membrane. In this outer membrane, you'll have porins. These are these trimeric structures I showed you. So they have holes that allow soluble molecules to diffuse across the outer membrane. You know, glucose, amino acids, they can diffuse across the outer membrane, get into the periplasm, and then be imported into the bacterial cell. Now, the outer surface of the outer leaflet is composed of lipopolysaccharide. So it's not just a normal lipid. And you also have something very important in the outer membrane. It's Brown's lipoprotein. So this is going to make a bridge between the peptidoglycan layer and the outer membrane. So functionally, it's important to keep these two structures attached together. Otherwise, you know, this could just float off somewhere. Okay, so something for you to think about for 5 or 10 minutes. It's not going to be on M clickers. So either on your own or with the people sitting next to you. Try and imagine it's a thought experiment. Gram-positive bacterium. How, you know, if you treat Staphylococcus aureus with methicillin, you want it to die, right? And then you can cure the person who's infected. But methicillin-resistant Staphylococcus aureus, MRSA, is a big problem. So just try and imagine what the mechanism could be of this resistance. Okay? Talk about it for a bit. Some of these are correct, but they're the only ones that I could think of. So maybe there's something else you could think of, another possibility. So think about it, discuss this for 5 minutes or so, and then we'll see what answers you get. Do you think this guy has any ideas? You could ask him and find out. Especially for the last one, something else. Hmm? Yeah, I think that there's more than one correct answer, but they're not all correct out of those options. Some of them are wrong. So why are they wrong? Which ones are wrong and why? That's the question. Ah, okay. Yeah, you're right. Yeah, so this enzyme is active in the periplasm. That's in between the cell membrane and the peptidoglycan layer. It's got to have access to that part of the bacterium. But in gram-positives, in fact, it can just diffuse through the peptidoglycan, so there's no kind of other barrier, really. Okay, let's see. What other time do we have? Okay, has everybody thought enough about these questions? I will take your silence for a yes. Okay, so option 1, who says we could become resistant by expressing an enzyme that destroys the antibiotic. Who thought that was okay? Just put your hands up if you think it's good. Yeah, a few people. Yeah, that's true. Okay, a lot of resistant bacteria have penicillinases, and they destroy the antibiotic. Option 2, you don't destroy the antibiotic, but you just don't care whether it's there or not. So you get an enzyme that is not going to be inhibited by the penicillin or methicillin. So option 2, who thought that was okay? Yeah, okay, fair few people. Yeah, I think you're right. So these transpeptidases were traditionally called penicillin binding proteins. Okay, so they're in the periplasm and they perform this step in the biosynthesis of peptidoglycan. So, and there are different families of penicillin binding proteins, the different families of this transpeptidase enzyme. Some of them can become resistant. They just integrate a couple of mutations and that's it. The antibiotic doesn't inhibit them anymore. So this is also a pathway towards antibiotic resistance. How about this one? Express a porin that excludes an antibiotic from the periplasm? No. Anybody say yes? You just did that to please me, didn't you? Anyway, so no, it's not true. This will work for gram-negatives, right? So porins in the outer membrane of gram-negatives can be more or less selective. But it's not the case here where we're talking about gram-positives. There's no outer membrane, so porins don't enter the picture. The last one, evolve a way to survive without a cell wall. Okay, one person said yes, but come on, this is going to be difficult, right? Very, very difficult. So mycoplasmas can do it, but it's because they are parasites on another type of cell, so they can get cholesterol. But for most types of bacteria, this is not an option, right? So it may be a theoretical possibility, but it's going to be very, very difficult. Something else? Anybody have any other ideas? No? Okay, well, maybe there are, but I don't know either. Right, one last thing before we all leave each other for today. So one thing I do with ABT every year is a little kind of optional exercise called Microbe of the Week. So after every lecture, I will send out a question by email to all of you. It's going to be something to do with the subject of today's lecture, okay? And the idea is that you try and find out the answer and then send me the answer back by email. Now it's got to be written in English in your own words, okay? Because it's quite quick to do a Google search, you find the answer quickly, and then just copy and paste the text from some other article or Wikipedia. So if anybody, that won't count, okay? That doesn't count as a correct answer. So it's a competition, right? So what I do is the first person that sends me the correct answer, 3 points. All right? Everybody else who sends me the correct answer, 1 point. Sometimes there's a bonus question, or if you put in a lot of detail and it's really great, okay, a bonus point, you know. But basically correct, 1 point. If you get the bonus question, you can have an extra point. So week after week, we have this competition, and so people, you know, get the points. Now, let me be clear about this. This is not part of the evaluation for the module. It doesn't count for your exams or anything. So why should you bother to do this? Okay, well, I don't know, just for fun, learning about bacteria, learning something else. Okay, this is the main motivation. Second motivation, there is a prize at the end of the semester. I think last year I did first prize, second prize, and third prize. So you get there's something, okay? It's not money, but it's real, okay? You get a prize at the end of the semester for the top ones. Last point, and perhaps not least, if you want to have a mobility year, or later on if you want to go do a master's and you want to get a recommendation letter from me, I always like to say positive things about students, okay? So if you do this and you send in a lot of interesting answers, it helps me to write something positive. This student was very keen, very enthusiastic, wrote a lot of intelligent comments. It was an optional exercise. It showed that they're involved in the teaching, okay? So it's good strategy for you if you ever want to get a nice recommendation letter from me. Okay, so we'll start 8 p.m. I will send out Microbe of the Week 1 to everybody. So if you get on your computers, get ready to answer to get like the 3-point bonus. Okay? So thank you very much, and I'll see you next week.